Hello everyone. In this video I'm going to introduce you to AResCoN, which is a pipeline that utilizes other pipelines for cell segmentation and image analysis so that you get eventually a full map of all neurons across different planes. So let's clarify what AResCoN is and what it isn't. AResCoN is not per se a trained network that is used to detect cells. It takes the output from already existing pipelines like CellPose, which I'm using, or even StartDist, which is also compatible with most of with the bulk, let's say, of AResCoN. And you can use the predictions that you get from these networks and apply them to AResCoN, which then calls Fiji. It performs some extra measurements and eventually can filter out observations that you don't like and keep only the rest. So the way that it works is that first of all you need an input that comprises a number of planes. You can use AResCoN if you want for single XY 2D image, and that's okay, that's fine. But what you can also do is to use multiple images of a stack, and I'm not referring to 3D segmentation here, I have to clarify, but I'm referring to 2D in a sense segmentation with a small range of Z scale so that you rectify for the wiggly nature of the tissue in immunohistochemistry. So many times, for those of you who are experienced with immunohistochemistry, you know that if you want to capture a large area, you cannot really capture every single neuron or every single nuclei or whatever at its crispest version, for the simple reason that it's always because the tissue is wiggly. It is always the case that some neurons are a bit deeper, some neurons are a bit shallower, and this way you have dim observations across with some crisp ones. So first of all, you need to start right with a microscope that can capture a few planes. In my case, I have slices of 30 micrometers. I would say it's preferable if you're a bit lower than that, for instance 25, that would be great, but 30 is also fine. And I'm capturing several planes, depends, it can range from only 4 to 13, of 3 to 4, mostly 4 micrometers. Let's stick to 4. So the images that you're going to see today are practically images that have been taken with an AI sample finder microscope, and with this microscope what you can do is that you can designate from the start the region of the tissue that you want to capture, and then it will just take a lot of images, it will stitch the tiles, and you will get an overall representation of the tissue. I can show you, for instance, one of these images. So I'm opening Fiji right now, and I'm just loading a stack which comprises this kind of planes. So here we have 9 planes, as you can see, and let's start with plane 1, and you can see at the bottom that these planes actually, this part of the tissue is crisp at the earliest planes. You can see how clear this is, but at the same time you see that if you go above, it's rather dim. Moving, of course, the planes, the picture changes, so you have dim observations at the lowest part and bright observations, crisp observations at the highest. So the first step that you need to take, and let's also change a bit the brightness so you can see more clearly what's going on here. So the first step that you need to take if you want to fully utilize AResCoN and not just use it for a single plane is to take several planes, to image several planes. And that's gonna take a bit of time. With the AI sample finder that I'm using for 3 channels and for a single hemisphere like this with 16-bit images and 20x magnification, I think it takes something between 20 and 30 minutes. So that's a lot of time, and you need to consider if that's something you want or not, in case you want to go for fully capitalizing on the capabilities of AResCoN, of course. So you get the idea, I hope, by now. So the idea is that by using another trained network, in my case I'm just going to use Cellpose, I want to get predictions from all planes. Of course, it can be the case that you get a prediction, let's say, for that particular neuron, for that plane, and you also get a prediction for that plane, which you don't want, actually, right? You would just wish that you get the crispest one. But of course, when you start getting predictions, the first thing that you want is to capture as many true positives as possible. So at this stage, we want to capture a lot. We make the network sensitive, in a sense that it's sensitive to many different things that can be the real observations. And then there is a way to filter out this kind of observations, right? So if there was an observation, let's say here, there is an algorithm that AResCoN uses to filter out the excess, let's say, observations that come from overlapping regions that correspond to the same neuron, to the same observation. Here I should also say that you can use AResCoN both for detecting the whole cell, the cytoplasm that is, or you can just go for the nuclei with CFOS. With CFOS there is a pre-processing of the image that I'm also applying, because I've noticed that it's rather hard to get really trustworthy predictions across a lot of different images, a lot of different samples, even if you have trained very thoroughly your Cellpose network. At least that's my experience. It can differ in your case, but let's stick to this kind of signal that you see right now, which is Team Tomato, by the way. And without further ado, because I've made a large introduction already, let's open AResCoN and see how it works. I should show you first what's the input that you want to have, and let's start with the images folder. And here you can see that I've divided this folder to subfolders that correspond to planes. If you have just one brain, of course, that would mean that you should have only one image per folder. In my case, I have two different brains here. I can reduce that to one later, so we don't double the time of this video. But what's happening here is that in practice I have 9 planes, only 2 images. They're multi-stacks, they comprise 9 planes, and that's the way that you should put them across your folders. So you have the images folder and you have the ROIS folder, and the ROIS folder is the folder that includes the primary predictions, the primary inferences that you get by running a neural network like Stardist or Cellpose. I've used both, they're both good, I have to say. I would prefer, to be honest, Cellpose for cell detection. For the nuclei, again, I would say that in my case, Cellpose work better, but that might just have to do with the nature of my samples, and by no means I want to underestimate the capabilities of Stardist as well. So, having described that, let's have a look first. Maybe that's something that you find interesting, how to create stacks, how to create images from a stack, and then run a Cellpose network to get the predictions that we want to get. And these are the predictions that finally end up here, okay? So what you see here, and when I say predictions I mean inferences, is zip files that contain all the regions of interest, all the ROI files that we later on load to Fiji. So let's see how we do that. I'm gonna open Fiji and I'm going to load this image that you see right here, and this is a multi-stack image. I've opened it just before. So if you go to the search bar and type stack to images, you should get this command, and now we have all 9 stacks. And what I'm going to do now is that I will just save each one of these stacks. I will show you how to do the first one. I'm quite sure you're experienced with that, but that's okay. And then I'll continue with the rest. I'll just pause the video for a while, and then I'll continue with the next step. So to save, you can do Ctrl S, which I prefer, and we have our plane. Alright, and I'm just saving it as plane 1, and I'm going to do the same for all the rest of the planes. Keep in mind that this part here is important if you want to use AResCoN, which is the case here. So you need to make sure that when you divide your image to stacks, when you separate the stacks, you need to make sure that they end to plane 1, plane 2, plane 3, and so on. So let's do that. I'm saving this, I'm gonna save the rest, and I'm coming back. Alright, we're back, and I've saved all the files from the multistack into single images from 1 to 9. Alright, and the next step that we're going to do is that we're going to get inferences from an already trained model with CellPose. I'm not going to show you on this video how to train a CellPose network, but I'm planning to do in a different one. But for now, let's assume that you've already trained the CellPose network, which you can easily learn how to do if you visit the GitHub page of CellPose. What I'm doing right now is that I'm logging into my Google Drive, and I'm accessing a folder which is called Collab Notebooks, and in the demo I've inserted all of these images. Alright, I've uploaded them already. Keep in mind that when you upload images in Google Drive, sometimes it takes time to access them from Google Colab, which I'm going to use right now. So even if you have already uploaded them, it might take a bit so that Google Colab can actually access these files. So let's log into Google Colab 2, and I'm going to use this file, which I'm going to provide to you. This is a file that's mostly created by its creators, the creators of CellPose, and I have just added some line, I won't lie, that was with the help of some generative AI, to proceed to predictions. Anyway, so let's move. First thing that you want to do is make an account in Google Drive, Google Colab, excuse me, and then you want to mount your drive, your Google Drive to Google Colab. This is already mounted because I did it before. After you run this step, you go to the second one and you install all the dependencies that you need to load CellPose and use it for inferences. Keep in mind that I'm using the L4 runtime type, which is really good for inferences. It's also more expensive compared to some other ones. For the training of the network, I use the T4 GPU, but I'm not sure if it's really preferable. Just keep in mind that there are differences between training and inferences. T4 is slower in my case. It also gets the job done, but with L4, when it comes to inferences at least, it's quite nice. Third one is just running this cell, and after this cell has run, we will go down to this one. Alright, and what we're going to do here is that we want to change 3 paths in your case so that the code runs properly. First is the path that accesses the network that you have trained. In this case, it's the TTOM CellPose network that I have trained and I'm utilizing right now. The second is the folder which contains the images that you want to get the inferences, that is the predictions. And the third one is the path that will lead to the output, and the output is going to be some masks that we're going to convert to ROIs out of this platform. And we're doing so because if you're working with a large number of conditions and rows, like 5 or 10,000, which is not far from our case, you don't want to convert all these masks to rows inside this platform of Google Colab, because it will take viable time. You're paying for that, remember? So let's do it outside, and pretty much faster, trust me. You don't have to have this folder created already. It will be created once you run this cell. Just make sure that the path makes sense and it originates from the same parent paths, right? Otherwise you won't create anything anywhere, I suppose. Okay, so let's run this cell. And this will take a bit of time. I'm not sure if I mentioned already, but in my case, using L4, it takes around 1 to 2 minutes to get a prediction, including all masks, for a single plane. That would make it, of course, around 10 minutes for 10 planes. So I have, around something between 15, let's say, and 20 minutes for 10 planes. I have 9. So I'm going to wait a bit, I'm going to pause this video, and then I'm coming back with the results that we have and the conversion of the masks to rows. Okay, so the predictions are taken already. You can see this check icon here, which means that the cell has finished and it's run successfully. And it took me 11 minutes along with the loading of the network, so quite good, I would say. Let's find our output and let's download it. So where is our output? Of course it's here, correct? So let's go to outputs and look for the demo outputs. Outputs is here, demo outputs, here we are. I'm going to download all these files, and it might take a moment because first usually Google Colab is Google Drive is zipping the files, and then you can download them. And the next step that we're going to perform after we download them is to create folders again, plane 1 rows, plane 2 rows, and so on and so forth, and we will just copy paste these images to their respective folders. Files are downloaded already, and I am going to extract them inside a folder. Let's use this folder temporarily. Let's paste it on our desktop. Here we are. I've already created a directory with subdirectories inside which contain nothing. Let's just make sure that they do. I think it's fine, okay. And next step, as I told you, is to let's split the screen in half. I think this is going to make it a bit easier. To paste the images to their respective subdirectory. So another thing that I notice here is that based on the code that I have, there's a conversion to mask at the end, and we want to change that, and we can change that now. I'm using auto hotkeys, which makes my life a bit easier when I want to make conversions of names. You will need that anyway for some part of the AResCoN, so I recommend, it's the Power Toys. Is it actually Power Toys or auto hotkeys? Let's see. It's the Power Rename, so it's actually from the Power Toys. So if you have installed the Power Toys, there is this Power Rename option, and here you can easily remove parts that you don't want from files inside a folder. So I don't want the underscore predicted mask, so I'll replace with nothing here. I'm just going to do apply, and voila, now we have the clean name. And let's start moving them to the right subdirectory, and we'll do a check afterwards, because these kind of things it's quite easy to make a mistake. 7, 8, and 9. Cool. Let's see if it's done correct. 1, 2, 1, 2, 2, 2, 3, 4, 5, 6, 7, 8, 9. Awesome. So right now what you're having, so that we make things even more clear, you're having images with masks. So how they look like is like this. Alright, so what this is all the acquired predictions, and they might be more than you see if you 0 to 1, tac tac. Okay, so these are all the masks, all the predictions that were captured when we were scanning the first plane. And if you recall, it was these kind of regions that were bright in our initial image. Let's have a look at our initial image as well. You see, so it's mostly this region that has been detected, which makes sense because it's not that crisp, but certainly more clear compared to all the rest, and so on and so forth for the rest, of course, right? But we don't want the masks, we want ROIs. We continue with ROIs. So how do we do that? Now it's actually the time that we're going to open AResCoN, and let's do that. So this is the new pipeline that you see is actually the AResCoN that I'm developing right now, and I'm using Visual Studio Code. I would recommend that you do the same. It's a really nice software to use. And what we're going to do is that we're going to File, Open Folder, and then I'm going to find the new pipeline, which is actually the pipeline that I'm using right now. And excuse me, let's close the Telegram, and let's just open the new pipeline. I've done it already, so there's no need to. It will lead me here, and multiplane cell detection, which I'm probably going to change to something like main.py or AResCoN.py. At this point is multiplane cell detection, and this is going to be the main code that we run, and this code can call other modules that exist here. So I also want to tell you that I'm also planning to make an executable file, meaning that you won't have to run this in Python. At the end of the day, you will be able to run it just by double clicking a file, which might take a few seconds to open, but it will be the same thing at the end. So run Python file, and let's see if we will get AResCoN open, and we have the window, which is nice. In case you don't see the window, it can be that it appears on this bar. Hope you see that bar. Yes, you can see. So just click on that and it should be fine. So the first tab is a tab that contains some information that are related to microscopy. The max plane gap is probably going to be removed in the future, so don't care about that one. Bit depth, right now I'm working with 16-bit. I've tested many things with 8-bit here as well, and it works. So I think if you go for 8 bits, just declare it here. It might work, it might not. I cannot promise. What I promise is that it can work with 16-bit if you do things properly. And for now, please select Windows. I'm also going to work in the future to make this applicable for Linux. It's not going to be that hard. I just have to change a lot of paths because paths change between these two systems, and I was not aware of more modern libraries that can really normalize that for whatever system you're using. So yeah, number of Z-planes. Here there's also a note, and I want to draw your attention. So far I've been testing it with up to 9 planes, and in principle there should be no problem if you have more than 9 planes. It's just that we're shifting from 1 digit to 2 digits, and that should be no problem if the code is written correctly, but I have to check. I haven't checked yet. I've just added a few more planes, but this is something that I'm going to check within the next days. But for now we have 9 planes, and if you have many brains, right, that they're consisted of different number of planes, of course what you want to do here is that you want to indicate the maximum possible number of planes. So there might be a brain with 4 planes and a brain with 9 and a brain with 12. If 12 is the maximum, then you do 12 here. Otherwise, it's not going to go across all your images. It will stop at plane 3 or plane 4 and so on. So 9 in our case. And then we have some main inputs that you have to choose, and this is I believe quite intuitive. So at first you need to select the images folder. By the way, I'm going to delete this. No, I'm not going to delete this. Yes, so you can select the images folder, the rows folder, and so on. But for now, let's jump to the change rows, because what we want to do right now is just convert labels to rows, as you can see here. So that's what we have, that's what we want to make. So for that, if I click now, you'll see that I'll get an error. It says that you have not defined the path to your Fiji file. Go to Measure Rows tab and select your Fiji.exe file. So Measure Rows tab, and this is the button that we have to click, and from here I'm going to navigate to the Fiji, and I'm going to provide you with a particular version of Fiji in order to make sure that things will run as expected. So I know that this file is actually this executable file, and I'm not going to click anything here as in generate measurements or anything. Even if I click, I should get errors because I haven't prepared for that step. But this is all I wanted to go to the change rows. Now if I click here again, we get this message saying that you will be now prompted to select the main row directory containing all plane N rows folders for conversion. The structure has to be the same as the one indicated in the main input tab rows, and the structure is the one that I showed you. Okay, let's see again. This is the structure. Okay, so this is what you want: plane 1, plane 2, and so on. So let's find it and let's select it. We're on desktop, rows folder. Here we are. Now I haven't opened Fiji myself, okay, because I've indicated the path of Fiji. AResCoN finds this path and it's going to run commands to Fiji. Okay, so I'm not typing anything right now, and AResCoN is just going through the batch process properties of Fiji, and it's inserting the right code to convert masks to images. And once it does it for one plane, it does it again for the second. However, the one of the ways to do that, at least, is to turn off close image J and open it again. Why? Because if you do so, when you finish with all the files that you have in one plane, it's a good signal to AResCoN that, you know what, I'm done right now, so you can continue with the next batch. This might not be the perfect way, but it's a way that works sufficiently, and after all, it's a matter of a few seconds till you open Fiji again and you type the command, and imagine that you're going to have 9 or 10 planes, let's say, right? So really, the repetition of the Fiji turning off and on is not going to cost you any significant time. The time that it's what is mostly heavy in terms of processing is things that are being processed. But right now, Fiji is very, very good at converting masks to rows, and that's what we're doing right now. And we're already in plane 4 or 5, if I'm not mistaken. We can see now. Let's see what it says. So plane 4. Okay, it's gonna take a bit longer, but we're getting there. And yeah, eventually we'll get, as you can see below, right, the rows out of the masks. There is a slight difference between the rows that you get, and this is actually something that in particular circumstances can be important. At least the way that I'm running these fidget codes, there is a slight difference between the masks that you get from Cellpose, I believe, and the rows that are created, because I've noticed that if I get the masks from Cellpose and using Python turn them to rows, which is the files that Fidget can understand, but I do it using Python and not using image.j, at least the way I tried to do so, there was always a gap. Imagine you have two adjacent neurons, nuclei, whatever. Cellpose, at least the version of Cellpose that I'm using, is never overlapping two observations. It can discriminate between two different objects, but it will draw contours that are not really overlapping. However, when you do it in Python, there's always also a gap left in between these contours. Small gap of pixels can be just a line of single pixels that separates them, whereas what I've noticed in this case is that when you convert to from masks to rows, if the observations are adjacent, it could be that there's really no gap in between them. The contours are super adjacent with nothing in between them. They're not overlapping in a sense that one shape is getting into another, as they do in Stardust, for example, but there's no gap. And that might sound trivial. However, there is a particular function that we're going to use later for detection of the relative background. You'll see what I mean. And the truth is that in that case, it's slightly better to have this gap in between observations. However, it's not something that we cannot, resolve. So it's really okay. Okay, last plane, which is good. Let's wait for it. And when you will run this program, I'll make sure that there's also a popping window here telling you that this particular procedure has been completed. I have it for most of the functions, but I've omitted to write one in this case. But this is actually done, and now if I go to the same folder, there will be another folder created which is called rows from labels, and here you can find the zip files. Okay, so let's have a look. Let's use another plane this time. Let's use plane 5 for instance. So input images folder, plane 5. So I'm loading the image, and then I'm loading Cellpose. Okay, so you can see that in parts that are clear with on this particular plane, Cellpose is actually quite good. You can see that some things are not detected. For instance, it doesn't understand that there are probably two neurons there, probably, and it cannot capture some neurons there. However, that's the very reason that we're using many planes. What we want to do is utilize observations from different planes and create at the end an overall set of neurons that correspond to all possible observations that have been taken, and we know that they're real neurons, right? And at the same time we want to filter out things that we don't like. So as you can see, Cellpose is quite robust already. I think that's quite apparent already. You can see how well it detects when you have something clear, and when you have something that's not clear, don't worry, because it's going to be captured on another plane. What we also are going to do with AResCoN is that in the end we're going to keep the crispest version only across different planes, which is quite cool if you ask me, and that's what I wanted to achieve in the end. Okay, cool. So now that you have all of these observations, you make sure that you take this folder and you place it in another folder. In my case it's called input. I've done it already, so this folder is pretty much the same with that one that you see there. Okay, so because I'm a bit compulsive when it comes to clear desktops, I'm going to get rid of some files that we don't need anymore. And now let's continue with running the rest of steps. Microscopy has been done already, so we're going to main inputs, and I'm going to select the images folder, and let's see how the images folder look like. You can also see here, right, you have the images folder, and inside you want plane 1 images, plane 2 images, plane 3 images. As far as I remember, it plays a role if you have plane n underscore n images, so please stick to the same naming, at least for this part, which is very important, plane 1, plane 2, and so on. Okay, so this is the input folder that we want to select. So we go to select images, desktop, input, images folder, and now it's selected. And then we're going to select the rows that we've created, and now it's selected. What we're going to do next is that we're going to take some measurements. For those of you who've used Fiji, I'm sure you know how this works. It's as if you're typing Control and M. It helps you get measurements, and what is important here, and I'm drawing your attention on that, is that you have already set your measurements on Fiji. So if you type set, not your measurements, set measurements, and you open this, you can see what I've selected, and I will make sure, actually that's a good thing to write down right now so I don't forget. So make sure that the measurements are set correctly. Okay, so these are the measurements that I want you to select. Actually, not all of them are really of use, but please select them so that we're on the same page. And after you do that, close Fiji. Alright, you don't want to have Fiji open, and let's do the rest. You can see that this is ticked. I don't know if it's a word in Greek, but I'm not sure if it's a word in English. But this is tick the boxes, so I guess it's fine. So we go to measure rows and we save folder. We need to select the save folder. There's no save folder right now. This is something you want to create, and I will explain what this folder is about. Let's just name it save, okay? And okay, we select it, and it says select folder to save your main row measurements. So once we get our measurements using our rows, the measurements for every particular plane capture that we have are going to be saved in a respective Excel file. You will see in a while how this works. We have already selected Fiji from before, so that should be okay, and we're about to hit generate measurements. You can always, before you click, go to this neuron information icon and see what is the function of the particular button. So it says that it generates CSV files, which is what we want to do. In the main save folder, these files contain the measurements of rows. No measurement data is saved in AResCoN at this stage, which is important. Okay, what we're doing right now is that we're creating Excel CSV files. That's all, right? Nothing else. Okay, I'm going to click on generate measurements, and just like before, I'm not doing anything right now, and it's going to run 9 times again and get the measurements of all the images. So imagine that you had more than one brains right now. You had something like 30 brains, right? Fiji would turn on and off again, not 30 times, but as many times as the maximum plane that you've selected is, which could be something like 9 or 10. So please always read the small box with the instructions before you start. So the names of the subdirectories have to be as I indicated. There shouldn't be an open instance of Fiji. I've also made some error handling, so in case you try to open, hopefully you should get, and I think you should get actually, a box telling you, a window telling you that you cannot perform this step until you first close Fiji. What is important is that it is not unlikely that if you have any kind of window which is open and includes the word Fiji, like a text file for instance that includes the word Fiji, or a PowerPoint or whatever, it might be detected. It's not unlikely that it can be detected at some point of this pipeline, if not in this initial check step, as Fiji which is open. So please don't leave any other windows that are open, because the way this pipeline works, right, it needs to know that it is the pipeline itself that opens Fiji and closes Fiji. So there needs to be only one Fiji instance open at a time, and that's what we're doing right here. So we've reached already on plane 4. Let's look at step 3. It says that you have to make sure that image.j will not ask for an update. For that, just open Fiji once and before you run AResCoN, and make sure that no update is asked, because this happens the very first time you open Fiji on a day, to the best of my experience at least. You can even untick the box of updates, because really you want a stable version of image.j in this case, and this is the version I'm providing. If you change versions, it's very possible that AResCoN won't run properly, because there are slight differentiations between versions in terms of where a particular button is, right? And the whole the way AResCoN works here is that it has a predetermined set of steps, like press tab 5 times and then click on the button that you will end up to. And if the cancel and process button, for instance, are vice versa in another version, which happens, then this is not going to work anymore. So please use the version that I will provide to you. Alright, let me see at my sticky notes and see if I have anything else to recommend here. Yes, one important thing is that right now the time that Fiji takes to open is predetermined. I think I've set it to something like 15 seconds or no. However, many parts of the pipeline are adjusted so that once Fiji closes, it turns on fast. However, you see that there's a gap. You notice here there is a gap until the word macro is written, and that is the gap that I deliberately leave there, temporal gap I mean, so that slower PCs can take their time for Fiji to open until the rest of the commands is written. There's not, or at least I haven't made a way that the macro and the rest of the things that are typed will be typed there after Fiji is completely open. The problem is that when you open Fiji, it already is indicated, in a sense, submitted, call it however you want, in the processes of the task manager. So there's not, or at least I haven't found, a very reliable way to overcome this problem, and the way I resolve this problem is that I leave that gap between executing Fiji and typing on the search bar the macro command or whatever. So in case that your Fiji takes more time, it's possible that the macro will never be typed on the search command because your Fiji takes more time to open. and for that we need to change a particular line in the code. We can have a look after that. I will shortly search for that, but before we get there, let's continue with the pipeline right now. So measurements obtained, new CSV files for each plane and image have been created, and you see the path that they have been created at. Every additional measurement that you're going to take from now on will be added as a column to the respective CSV file of the corresponding plane and image. Let's see what this is all about. So I'm going to the input, and this is the save folder. And remember, we had 9 planes and 9 images. It could be that if we had 100 different brains, it would be 100 different images per plane folder, right? And I'm opening it. And now what you can see here is pretty much all the measurements that we've set for all the rows that we've obtained by running the CellPost network. So this starts to be fascinating in a sense, because we have an automated way to obtain all information that we want about every single observation that we've made. So this was the next step, and now that we have our measurements, remember, these measurements belong to your PC at this point, but they don't really belong to your AResCoN, and you want to add them. So I'm going to click add measurements, and what this is going to do is to incorporate the CSV information to AResCoN. At this point, for instance, if you had closed your AResCoN program, it should still be okay, because imagine you open it again, right, and you want to add measurements. You will be asked to indicate again some of the folders. So let's start by selecting the main save folder. Normally you wouldn't get that, but I actually closed it myself. You never saw it in the video, but I closed it myself and I opened. So let's click OK, and let's indicate the main save folder. And the save folder, by the way, please keep it the way I have it. Keep your save folder inside this directory that contains the images folder and the rows folder, okay? Because sometimes the save folder is used for the AResCoN to understand where the rest of the files can be. So please keep it here. So we're selecting the save folder. And now, in principle, if you go to add measurements, okay, you have to select the main row folder, of course. So let's go back, and this is the main row folder. Let's see if it will ask for anything else. No, that's fine. It might take a while. I'm also planning to insert a bar for that. In this case, it didn't take much time, but imagine that you have a lot of images. It will take some time. It might take up to I don't know, like 20 seconds probably per brain, meaning the whole set of the planes that it has, which translates to a few minutes if you have more of them. I'm trying to figure out ways to make it even faster, but I think it's working quite sufficiently, at least for these demonstration purposes, and not only, of course. So step completed. All measurements are now added to AResCoN. Consider saving your new measurements to disk now. In case, let's say you've added your measurements, it took a few minutes, and you don't want to repeat that procedure, but you want to close AResCoN. So what you can do is that you can save your measurements, and I think saving is what takes actually a bit longer, not adding the measurements, but saving. And for those of you who are into programming, the reason is that, and I would appreciate some feedback, I made a class in a different module, and I cannot save directly a pickle file by the module. It has to be on the main code. So I'm converting the whole class information back to dictionary, and that takes a bit of time. So any feedback here would be deeply appreciated. So I have saved successfully the pickle file, which says a pickle file is a file of Python, in the sense that retains information that you can load later on, and it has been stored in the workspace directory. Let's remember, so yeah, the workspace directory, and this is important, is this directory is the directory where you run AResCoN. What this means, of course, is that if you run different datasets, things will be created, new files will be created at the same folder. And I'm raising your attention here again because you don't want to mess with different workspaces. So imagine that you want to work with a complementary dataset later on, or a different dataset. It's easy. You go to AResCoN, then you copy the whole thing, you paste it, you make sure you delete, and I'm going to include a button for that actually to delete all unnecessary files, so you make a fresh start. So right now you can see the saved metrics, and this is the file that has been saved, and this contains the information of our measurement. So let's try closing AResCoN and opening it again. Now if I load measurements, it says the data is now passed to AResCoN. It can detect the data from the saved metrics and it can load directly. In the future, again, I can change that so that you decide which is the particular file that you want to open, and this is more appropriate. But for now, please be careful that you want saved metrics that correspond to your real metrics, and just make sure you're not opening previously saved things. Okay, now that we have that, what we want to do is to add new metric columns to our CSV. Which ones? Well, the entropy, it's the Sauron entropy that I was trying to get when I was really trying to find a way to discern and locate the crispest version of neurons across small z-range. It's not useful anymore, at least for me. You might be working with datasets that entropy makes sense to study, so I decided to just keep it there, just to know it's going to take a bit of time. If you want to run entropy, especially if you have large images with a lot of observations, good luck probably. So let's forget about the entropy and let's go to the fine edges, which is something that we need. It's something that we need especially for the Z filtering. So if you're working with images with a single plane, you can still use AResCoN. You just use the first plane n. Okay, so you have to simulate this, and you can put all your data in here, and that's fine. All right, and then you go to microscopy, you select number of planes 1, that's fine. I do 9 in this case. However, if you want to obtain information from all different parts and then to filter the overlapping parts, you need to do the fine edges. And actually, before doing the fine edges, I think this is a good time point to show you how predictions look like when you collect multiple observations from multiple planes, so it will become more clear to you why we need to do the filtering. Alright, so I'm opening Fiji and I'm going to open the whole multi-stack of images for this image. And what I'm going to do next is that I'm going to load a lot of rows. Okay, and that's really a lot of rows. Actually, it's even becoming heavy for my computer for some reason. Yeah, something has lagged. I think you can still see me, but my computer is struggling right now. I don't know why. Let's try to overcome this problem. I'm going to close the Windows Explorer. Now maybe restart. Okay. Try to refresh our image. Okay, sorry about that signal. It is annoying. But let's continue. So loading the third, the fourth. Oh, it's having trouble again. That's interesting. the fifth. Okay. So now we have a lot of rows. Okay. And you can also see what the problem is. So you will be wondering why why in the world do we have all these observations across clearly, apparent neurons? Well, the reason is that when you start going away from the plane with the clear ones, things start to get messy, right? So the observation that you see there is actually an observation that comes when you have that particular plane. And there's nothing valuable in that particular plane for this part, but there might be, for other parts. And this is the reason that you need to apply the filtering, right? You don't want double rows or triple rows or even worse. And that's why we're going to do the Z filtering later, and for that we need these find edges. And what the find edges is, we can also have a look at that, is a way, is a Laplacian equation that separates, in a sense, the contours of it doesn't know that it's the contours exactly, but I mean, I don't know the details, but it can understand the difference. It can understand the difference in the contrast, and it can accentuate things that are potentially contours. So if I do find edges, let's do this for this one, right? You see that now it has converted the image, and now if we have a ROI, and assuming that the ROI is this one, and we take a measurement of that ROI, then we can take its standard deviation, and the biggest the contrast is between the contours and the inner information of pixel values, it probably means that the more crisp in reality the neuron is, because what makes something crisp is actually its difference from the background, right? And using the find edges is a good way to detect this. So that's what we're going to do. But because you can probably see this icon, which really gets on my nerves, I'm going to pause this video and I'm going to start a new one after I probably restart my computer. We're back from where we stopped, and now, as you can see, there's no blocking icon or something, so we have a clear screen to continue with our tutorial. And let's continue with find edges. Okay, so I've loaded the measurements again. Just to remind you, when you load your measurements, you're actually calling this file, which has your measurements inside. And now we're going to find the edges. So this button, what it's going to do, you can also see, is that it will run Fiji automatically, as always. It will execute the find edges command, and it will just take all measurements again, and from these measurements it will keep the standard deviation, and it's going to be a different standard deviation compared to the initial ones that we've taken, because now we've applied this image analysis procedure that changes, right, the value of the pixels and accentuates the contours. And what we're going to do later on is that we're going to make sure we're selecting the most crisp version of a neuron based on not only based on that, but taking also as a criterion the standard deviation that has been taken from the find edges. I hope by now this is more or less clear the way it's happening. So let's click that button, and Fiji should open. And as always, we are going to wait a bit. We'll see together a couple of times how this works, and then I can pause the video again and continue after all the measurements have been obtained. So what I want to emphasize on right now is that at this stage you're just creating CSV files that correspond, of course, each one of them corresponds to a particular image of yours and the measurements of the rows that they belong to this image, but nothing else at this point. Okay, so you're just creating new files, nothing else. And the next step that you see next to the find edges, which is the add find edges, we'll see in a few moments after this window goes away. Okay, you see these add edges. So these add edges, what it's going to do is that it will take the standard deviation measurement that has been taken from the find edges images, and it will insert it as a separate column to the initial safe measurements that we had to look at before. I think you remember these measurements. These were the measurements that were taken by implementing the rows to the original images. This is just an extra measurement that we will incorporate into that CSV file. So what the find edges do, once again, is that it just creates new CSV files, which eventually we're not going to use. What we're going to use out of them is particular columns, that is the column of the standard deviation, that we will correspondingly add to the respective initial measurements that we've taken. So when I say initial measurement, I refer to the measurements that we took in this tab, in the measure rows tab. And yeah, this is just terminology. Where are we at right now? Let's have a look. I think we're something like plane 4 or 5. It's also recommended not to click on many things when this thing is running, of course, because what AResCoN does is that it's using a library, two libraries. One of them is called PyAutoGUI, and PyAutoGUI practically drives the clicks on your PC. So if you also start clicking at the same time, this is going to be problematic because it might interrupt the procedure. That's why I ask you to take your hands off your mouse and keyboard as long as this thing is running. It's the safest way to go. And PyAutoGUI is sometimes a bit problematic in terms of activating the images and clicking properly. I've done a lot of workarounds to make sure that it's going to work successfully. I hope it will work in your case because it's been tested thoroughly, so that it runs without problems. However, if it doesn't, for whatever reason, you might need to interrupt, because imagine that for whatever reason it cannot activate your Fiji panel that you see right there. The rest of the information regarding the steps that are going to be performed still are running. So as you can see now, tab is pressed, Control A is pressed, delete is pressed, Control V, actually not delete, is pressed, and so on. So imagine that the Fiji gets stuck, all these steps will still keep running on your PC. So if you start opening folders, it could be that Control C is pressed, and this can really mess up with things that you're doing. So if something is going wrong, and don't worry, because there's not possible case that a click will be made by itself and files will open if something goes wrong. It needs, your intervention is needed for something to go wrong. But in case something goes wrong and you see that the whole code has stacked on Fiji because it didn't, Fiji took more time to open than usually, for instance, something happened to your PC, and within the first 15 seconds, that is the leeway of the Control L to be pressed here and macro to be typed is passed. The best thing that you can do is to terminate the whole procedure, and you can try clicking X on AResCoN, or you can try opening the Visual Studio Code and killing the procedure from there. I will show you what to click. I'm not going to click it, but what you would want to click is right click here, kill terminal. If you do that, the code will stop running. Having said that, if you stop running the code and you've already initiated the batch process macro series of steps in Fiji, then Fiji goes by itself and it continues running. So you have to close Fiji too. This is just a precaution in case something goes wrong, but don't be concerned. It's highly unlikely that it will mess up with your computer, as long as you're not, when this is happening, you're not really playing with your most essential files or something. But everything should go well, really. I'm just warning you in case something goes really wrong. Just kill the terminal and it's fine. So the measurements are obtained, and it says the standard deviation measurement following Fiji find edges has been added to this path. Let's see what this means, okay? So you see a new folder is created. It's called find edges results, and what we can see here is that it's a set of measurements just like the first ones, only that these measurements are obtained following the find edges conversion, and what we want to take is this, nothing else. And where do we want to paste? Where in this case we have plane 1 and this image. We want this column to be added to this column. Alright, so we want a new column here with the new find edges, with the new standard deviation based on the find edges, and we want the same thing to happen for all of them. So what we're going to do is that we're going to add edges, and now it's the terminology I told you, it's a bit complicated, because add measurements, what it does, it's bringing measurements to AResCoN, whereas add edges, it's bringing measurements to a CSV file. Okay, so it's a bit different, but I'm going to add edges, and it says it's recommended that you create a backup of your measurements, because I don't know, maybe something can go wrong, so I decided that it would be good to create some copies here. And in this case the backups are created pretty fast, and you have the backups of the save, the initial measurements, and the backups of the find edges results that we just looked at right now. And it says the metric standard deviation has been passed as a new column to the initial measurement files. Let's verify that this is indeed the case. I'm just going to go to another one, okay? I'm going to plane 4, but it's happened everywhere, and you see we have a new column there, which is exactly what we wanted to do. So once we have done that, now remember what the add measurement does. The add measurement reads the CSV files and adds the information to AResCoN. Because we have changed the main measurements and we have added the standard deviation, we need to add measurements again. Okay, so that's what we're going to do. And these new measurements will include the column of the find edges std_dev. We have it. Why not saving it as well? It wouldn't hurt. It takes a moment. I guess it's fine. As I told you, if you have any feedback, I will appreciate to make this a bit faster. And now that we have it, it's time for the next step. And this is actually the final step if you have just a single plane, and that's all that you want, right? A single plane, many images. You can run this final step now, and you're pretty much done. So what we're going to do here is we're going to take some relative background measurements, as I call it. And what this is that imagine that you have an observation, a prediction of a row here. What you can do is just set a crude filter saying that if the intensity, that is the value of the pixels, the mean value in terms of intensity of the pixels comprising this row is at least a number, then consider it an observation, otherwise do not consider it. This is very crude, a very crude way to go, and I used to go more or less that way in the past. I would take a mean measurement of the whole image, and then I would compare the difference between the mean value of the whole image to each single row, and I would filter in and out, and then manually I would intervene to sort of rectify what was wrong. And of course there will be mistakes, because when we have a large image, it's really hard to get a representative mean of a background noise. So what we want to do is to take a relative background, a background that is an enlarged region of the initial measurement that you have, 30 times, 40, 50 times, whatever. So it's a small, relatively small region surrounding your cell, and this is more trustworthy as a means of background. But of course, if you have regions that are densely populated, the next challenge that you're facing is that you're including in the calculation of the mean intensity parts that are not really the background. Okay, it's just other neurons. So this is the reason that is important. Either you're studying the cytoplasm or nuclei, especially with nuclei, I would say, because with tomato I'm pretty much including every possible detection, but especially with nuclei, you really want to filter based on a background noise, right? And to do that, you really want to have as many observations at the first stage as possible. So you need to have a lot of positives, either they are true or false positives, but it's good to have them there because they are masking, in a sense, parts that you don't want to study. And this is what's happening right now, and that's the reason I'm elaborating on that, because I think it's essential and it's a good step towards progressing and achieving higher and more successful filtering. So what the macro is going to do here is that imagine that this is the region that you're going to take as a relative background. Anything that has been detected by CellPos as a candidate region of interest, just like this, will be taken out. So for every cell that we have, the cell itself and the adjacent cells that have been detected by CellPos will be taken out of the equation. Okay, so we're just including all the other background information, excluding the rest. And this is one of the most trustworthy, I would say, ways to get a really meaningful background. So the raw enlargement factor that we can use is something, we can use something between 30 or 40. I will do 40. And the next two steps, they have to do with the it's either auto hotkeys or power toys. I'm not even sure if it's the same thing. I have to check. Let's check actually right now. It's a auto hotkey, as you can see here. I'm not sure if it's originating from this application or if it's a different one. I will clarify in the documentation anyway. But you need this kind of program. You need to install this program in your PC. Why? I will also explain. And again, it might take a bit of time. I understand you might want a faster tutorial, but I think it's good to have everything included in this one, and then of course you can filter out the information that they're not that valuable to you. But just to get into a bit more technical details here, there were some occasions that while we'll be running this filtering script to get a relative background, for some images an error would be raised, and this was not an important error, but it needed the intervention of the user, so the user had to click OK for the Fiji to continue running. And because PyAutoGUI as a library was already running there, including it as a second time to press enter when this message appears was rather problematic for really technical details. So the reason that the way that I managed to overcome this problem was to just incorporate another software that pretty much can do the same thing. Operate your screen, perform some steps. So what I'm going to do here is that I'm going to locate a particular script which is inside the workspace that just presses enter. Okay, that's all it does. Actually, if you see, this is a very simple text file inside, and then you want to locate the executable file of the AutoHotkey, and I'm using AutoHotkey UX, and this is all you need, and this will just call it when it's needed to do the dirty job, I don't know, of just pressing enter, that's all. So that you don't have to do anything, right? That's the whole purpose of the pipeline. So now we're ready to start getting the ROI background mean gray differences. We've set the enlargement factor, and we've selected the script and the executable file of AutoHotkey. As always, make sure Fiji is not open, and then just click. Now you're prompted to select a directory where the masks will be saved during this image analysis, and for that we have the masks directory. I created this directory inside the input folder, of course, and I'm selecting it. This is just for sanity checks, nothing more. You can use it if you want or not. You'll see how it looks like. I hope I selected it correct. Let's see. And as always, Fiji starts running, and let's wait a bit. In a few seconds, macro will be typed again. That's right. Starting from plane 1. Beautiful. So, what's happening here, in this case, in this scenario, there is this error occurring, as I told you, and now the AutoHotkey is actually intervening, and if you see, there is a small part that's cut out of the image, and this is a convex hull that's created. It's created based on the edges of, let's say, based on the extremities of the observations, so that you get a shorter image, and that way we can sort of differentiate better the signal of cells that are really close to the background. Okay, so the convex hull helps us to get a more tight, let's say, prediction of the region that includes our regions of interest, of the larger region that includes our regions of interest. And let's see again how this is going to work. The error first, as I told you, sometimes it happens, but in a while you're going to share observations, and the convex hull will be created across these parts, and you can see that what's happening right now is that we're applying our ROIs, we're masking these regions out, and these rectangulars that you see there, these squares, is actually the regions that are enlarged. Okay, and we're taking the mean background of these regions, but we're excluding every white part that you see there, as I described earlier. So everything that's whitened is a mask, so it's not included in the estimation of the relative background, and the rest of the information is there. We can see one more, and then I'm pausing the video and I'm continuing after all the information is obtained. Let's go for plane 3. And as I told you, this is close to the final step, the penultimate, I would say, step for those who are working with one plane. So let's have a look again at how the image looks like. Now we're at plane 3. The error, as always, AutoHotkey intervenes, it presses enter, and the convex hull will be created along these lines. Here's the convex hull, and it helps, as you can see in this scenario, right? You're not taking the background that's outside. It's not possible to take the background that's outside the brain anymore, because everything else has been whitened out. that's the beauty of it. And now you see, let's zoom in, for instance, for this particular, let's say, observation. We have this square, so we're taking the mean background, but we're excluding this, and this, of course. And we're doing the same everywhere, and also for the cells that are close to the edge. This is not going to be included anymore because we've successfully managed to take it out. Okay, pausing the video and getting back after everything is updated. Nice. Now that we've obtained our background, ROI background results, we can see that if we navigate to the input folder, there is a new folder that has been created, that is the mean background results. Let's go, for instance, to plane 8. And what you're going to see here is, again, measurements taken, correct? However, these measurements, of course, are taken based on these masks that we applied, and you saw. So when you have mean here, it's the mean not of the ROI itself, but of the enlarged region that surrounds the ROI, and of course it's a mean that excludes other detections. Okay, and at this stage of the pipeline, this information is actually submitted automatically to the main measurement. So if you go to the main measurements here, and sorry, I've been using another file. I'm going to delete very fast these guys here. I sort of cheated because I passed another file that contains the information, so I don't lose time again, but it's not going to take much. Cool. So let's go to plane 7, for instance, and what you will see now is that apart from the defined edges that we've already taken, now we've added 2 more columns. One is the surrounding mean, and the other one is just the calculation of the difference between the surrounding mean and the real mean, okay, of its ROI. That's something you can also do manually, but it's good to have there as measurements if you want to apply it. So that's it, and that's of course the case with all measurements. And now that we have it, it's time to make your final filters across the X and Y axis. Again, I'm repeating, if you have single images, you apply this step and you're done. If you open your metrics, this just opens the first file in the folder. You can see what kind of metrics you have here, right? And you can apply your own criteria in terms of filtering. Now, because doing detection of cytoplasm and even a slight indication of something that is a neuron might be okay, at this stage I'm not really inserting any measurements related to the surrounding mean, but you can certainly do so, and if it was publishing a paper, maybe I would do so as well. So what you want to do is that you want to find your surrounding mean, okay, and you want to find also your real mean. So if you wanted, for example, to filter out some detections that are not bright enough compared to the background, what you could say is that mean is larger than the surrounding mean. I can see how exactly it's written if I just click here. Alright, surrounding mean, okay, times 0.3, for instance, right? And you can do the same with any other kind of filtering criterion that you want to put. For instance, you can say that area is, let's say, smaller than, I don't know, like 300 and larger than 10 pixels, and we can actually have a look, so we don't do something super arbitrarily. Let's open our multistack and let's see how much area a big neuron occupies. This looks like a quite big neuron. If it's just one, it seems like it's just one. And I will go a bit further than that, because it can also be that, you know, somewhere maybe across the image you have something which is even bigger. maybe a bit slightly bigger. Okay, now it's certainly big, I would say. So I'm going to just run a few measurements, and you can see that the area is actually 1078. I will do 1100, let's say. So if area is smaller than 1100 and larger than, well, 10 for sure, actually maybe we try to search for a small neuron in this case. Let's see, maybe this guy will do. Not really sure if that's a neuron there, but you know, we have small one there. Of course, you need to be a bit more careful when you apply your measurements, but for the sake of demonstration, that would be fine. So we have 62. I'll drop it to something like 45, maybe something like that. Maybe a bit less. Let's do 30, okay? I want to include as many as possible. And you can put any other filters that you want. So in order Arrow to pass the filters, all of these conditions that you write here must be valid. If this condition is not valid or this condition is not valid, then it will not be passed there. Okay. Another thing is that you can also insert and within these conditions and make them more specific. So this is the area, and I want it to be at least, you know, the circularity has to be at least I don't know, 0.8 or so. Okay, you can do that. You're making it more specific, so it's more likely that you filter something out, but you can do that, and you can put OR here as well. This is to make a set of conditions within one condition. But in my case, I will just leave the area, and I won't do any further. And I think the area for now is good for these purposes. And let's make sure that we add the new measurements, because now our main measurements have more information, right? So we want to add these measurements. Okay, now they're added, and now it's time to apply the filters. And one more thing, within one condition you cannot mention the same name twice, which is of course limiting a bit your options, but you can make factors, right? You can make it in the form of a factor if you want to use something twice inside the same condition. This is just the way it works, and it's quite technical behind. But of course, if you have different parentheses, right, different conditions, you can use the same measurement, the same metric there, perimeter, circularity, or whatever area, right? So all metric names have been validated, and these are the conditions that they would be applied. So again, if a neuron is either larger than this value or smaller than this value, it will be tossed away. Cool. Let's continue. Now we have to select an empty subfolder where we're going to save the accepted files, and we can make one here. We can name it filtered xy. All right, and I'm selecting the folder, and the filtering is going to take place. And I think in this case, I've also included a window, a message when all the calculations are done. You can also check by clicking at the AResCoN window. If it says not responding, it means that it's still running. I'm sure it won't take long. Okay, your filter rows have been placed inside this path, and it's recommended to create a pickle file. This pickle file is super important if you also want to go for Z filtering, which we're going to do. So yes, go for it. But before we go to the Z filtering, let's see how our predictions look now. So we have and we get a message that the pickle file has been created. Filter XY. Alright, you have the accepted and the rejected. The rejected ones are categorized, of course, according to the planes, and you can see the condition that's translated into accepted forms of naming a file. So this is sort of the condition, and we can see, for instance, what's been excluded here. Okay, let's have a look. I'm going to open, this is plane 3 if I'm not mistaken, yes. So I'm going to open plane 3, and now I'm going, let's see how many rows there are. Okay, it's like 22, and let's see where they are spotted exactly. So you see, tiny little one, big ones here. So these are all things that have been taken out. And let's see what's included now. Let's go for plane 3, and so we'll remove the previous ones. Always remember to remove the previous rows from the row manager. And did I close the file? No, I didn't. So let's see what's included. Okay, and as you can see, many of them are included and they look good shape as well. So far so good, and this is pretty much the end for those of you who are interested in just XY filtering. Having said that though, I'm going to apply soon a complementary part that will utilize other pipelines for region detection, automated region detection. There are some pipelines who claim to do that completely automated after you train a network, and some pipelines that require from you to send some landmarks. Not many, still some landmarks to get predictions of the regions. So my next ambition is to integrate this spatial information to the information of the polygons, that is the rows that belong to the hemisphere, and eventually, right, be able to also produce a file that not only contains the measurements of all the neurons in one hemisphere, but also to which brain region they belong to. And because when it comes to whole hemispheres, there are differences in terms of the intensity of neurons when you do a staining in different regions. What I also want to make feasible is to have a bit more flexible filters when it comes to the mean gray value and the surrounding mean. Saying, for example, that if it's the infralimbic cortex, I want the nuclei, right, to be at least 20% brighter, but if it's the somatosensory cortex, I want them to be at least 10% brighter, because I've noticed that for this particular staining that I'm running, different regions have less pronounced signal. But that's something that I'm aiming for in the very foreseeable future if I want to finish my PhD sufficiently, which I do. Alright, so we've had a look at this, and that's working nice. Final step for now, guys, and this is the Z filtering. Okay, I strongly recommend to not change anything in the Z filtering. What are all these things here? Well, they correspond to some variables, and I've made a decision tree, so if you want yourself, you can investigate. I'm also going in the future to insert a few images for what happens when you tweak these values. But if you want to investigate by yourself now, you can go to the decision tree, which is inside the workspace, and here you can see all the rules that are followed. First, to remove the engulfers, and the engulfers are all these dim, larger predictions that are made when you move across planes and you go from something crisper to something dimmer. The signal gets larger, so first you need to remove observations that hug, that engulf other observations, and then you end up with like cells that are not overlapping with any other cells, which is the ideal scenario, with cells that are overlapping with one or with more than one. and you can see the different colors here, like the cyan, like the yellowish, and the sort of beige, I guess. And this corresponds to the colors here, and every condition is named in Latin, so you can see, what condition corresponds to what and what happens when you change it. I can be more descriptive as to that if you ask me. It's quite complicated, so I cannot just remember things by heart. I have to go back to the algorithm that I developed and remember what I did. So recommendation, just keep it as it is, alright? And let's try to apply filters now and see how it works. So before proceeding to Z filtering, you need to indicate the accepted row is pickle file. And we know where this file is. Let's go to the input. We have the filtered, and we have the accepted. There it is, the pickle file that's created. We're going to open the file, and again, I believe that I included a message after the filtering is done. Let's just make sure that this is the case. Cool. So that's for now the final step. Let's see how well it works, and again I will shortly explain what are the future steps to be done. So your Z filter rows have been generated, and they are inside the accepted Z filter rows. Cool. Let's have a look. Now filtered, accepted Z filter rows. So this is what you want at the end, and this is what all the fuss is about: getting a complete set of predictions based on all of the planes that you have in possession. So I'm going to open the stack again, and I'm going to load the final file, and this file should include all the observations that we want to obtain. Alright, so let's start from these planes, right? And we know that things start becoming clear here, and now I'm going across the observations, and you see that they're more accurate than before, which makes sense because of course now it's including information from all planes and not just one plane at a time, right? And the beauty of it also is that you can see the plane that they're originating from and how well the estimation has been made. So this dash and the number 4 indicates that this is originating from most likely from plane 4. And if you go to plane 4, you have a crisp image. You go to plane 5, it's not anymore. You go to plane 3, it's not anymore, right? And you can see that some are on plane 5. You go on plane 5, indeed, it's a bit crisper, right? Compared to plane 4. Someone could argue at least. Sometimes there are, it's not extremely accurate, but most of the time it's very accurate, which is really nice to see, working so beautifully. And you see all these label names. So let's go here, for instance, which is a bit more confusing region, plane 7, and let's see how well the predictions are made. So image, adjust, brightness. I'm going to the challenging parts now, as you can see, and let's see what we have, and look at it. Indeed, it's working beautifully. Again, if you don't like these kind of things, right? I don't personally like this kind of thing, or this thing. It's very marginal to be considered. But with the filtering, you can easily get them out. And this is the filtering of XY that I showed you before. You can play on that, and you can filter things that you don't like if they're not circular enough, if they're too big, if they're not that intense. This is all up to you, really. And let's look at some other images as well, originating from 7 and 8. Indeed, let's find an 8. For instance, this says it's 8. So let's go to 8. Okay, yeah, it's it starts to shape, but it's not that apparent. Here it says it's 6. Let's go to 6, and indeed, oh yeah, here comes the 6. So you see, that's the power of this pipeline. So it is utilizing predictions from AResCoN. It's not really a 3D prediction. So I tried a bit to use the 3D prediction from different pipelines. It didn't really work, and it makes sense because it's not that you have a complete 3D image here. You just have slightly different depths, which are most related many times not to a depth of the neuron, but to a depth due to the wiggly nature of the tissue. And applying AResCoN, you can eventually get a very nice final estimation of neurons. I'm just showing you around, right, how well it works. And this is, of course, also due to the robustness of cell pose. Without cell pose, that wouldn't be possible. I also want to emphasize that it's likely that if you use StarDist, based on this pipeline, I cannot guarantee, I haven't tried it yet, if you can get really good, or if you can even get the Z filtering, because a difference between StarDist and cell pose is the fact that you never have overlapping on the same plane, overlapping observations, and that plays a role later on. So just to keep in mind, let's just look a bit, a bit around, because I'm always very excited when I see how well cell pose works, of course, and how well AResCoN incorporates it to the detections. And I have to say, of course, that this is an image that wasn't used during the training of the model. This is clearly a test data sample, and it's just working beautifully, right? Let's just go to a bit dense regions, see what the labels say. They say go between 3, 4, and 5, and indeed it's around 4, most of them, and you can see how beautiful the detections are. So this is AResCoN. I hope you like it, and I hope you'll use it for your projects. Of course, I would deeply appreciate if you can cite me, if you can cite the GitHub page of AResCoN. This is, of course, a work that took me months to achieve, and it's going to incorporate more properties, of course. As I told you, one of the goals is to include these estimations of regions, again taking outputs from other pipelines. And what I also want to do, and that's going to be done very easily because I've done it in the past in other pipelines, is in this case that we have 2 channels, to also get the overlap. That's something that can be done within 1 or 2 days, to get the overlap based on a percentage that you dictate of these 2 channels. And then you can have observations, neurons that were expressing both CFOs and both the tomato, for instance. That's going to be done very, very soon. So this is AResCoN again. Please cite me, and I have to tell you that I'm looking for a job in Australia. I really love Australia. So if you know anyone who's interested in these kind of inventions, I'd be glad to get in contact with them, because my dream has always been to go there. I've been there twice and I just love it. So cite please, and let people know about this kind of tools. You can also check the EasyFreezy, which is another pipeline that I've developed for detecting freezing behaviors based on deep lab cut. And I'll stop advertising myself because it's mostly about the tool here. So have a look, send me a message. I can certainly help. Just to let you know, I'm in the last months of my PhD, so things are quite intense, but I'm always happy to help, and I truly believe that this is a pipeline that has been carefully developed, and it can help you solve this kind of detection issues. Again, a lot of credits to the people who made cell pose mostly, and of course all these people who are contributing to advanced 3D, because the only thing that I'm really doing is just bridging a gap between cell detection and some further processing and analysis here. Thank you very much, and we'll be in touch soon with the newest updates of AResCoN. This is going to happen.